We are often told that proteins need a specific folded shape to function properly. In biology classes, we learn that a protein's function depends on its three-dimensional structure, like a key fitting into a lock. But here's the puzzle. Some proteins do not fold at all, yet they still work. And in fact, they can be essential. How is that possible? These are called intrinsically disordered proteins, or IDPs. Unlike the tidy, folded enzymes and structural proteins we're familiar with, IDPs are floppy, flexible, and don't have a stable structure under normal conditions. That might sound like a broken protein, but it's not. These proteins are intentionally disordered, and their lack of fixed structure is what gives them a unique advantage. But why would evolution keep something that seems so unstable? Let's consider what flexibility means. A structured protein can perform a very specific task, like cutting a chemical bond or carrying oxygen. But a disordered protein can shift its shape and bind to many different partners. It's like a shapeshifter. It can adapt to different environments and roles, which is especially useful in signaling and regulation. Cells are dynamic environments, and IDPs are perfect for multitasking. For example, take the p53 protein, one of the most studied proteins in cancer biology. It helps control whether a cell divides or dies. The disordered regions of p53 let it interact with dozens of different molecules depending on the cell's condition. If p53 were rigid, it couldn't do that. It would be locked into one function. Another famous example is alpha-synuclein, found in nerve cells. It is disordered under normal conditions, but it can bind to membranes or other proteins when needed. However, under stress or mutations, it can misfold and clump together, and something seen in Parkinson's disease. This tells us that being disordered can be helpful, but it also carries risks. So, what does it mean for a protein to be functional without folding? It means that folding is not the only way to create biological activity. Disordered proteins often become more structured temporarily when they bind to a target. Think of a scarf. It's shapeless when draped over a chair, but you can wrap it tightly around your neck when needed. IDPs do something similar. They conform just enough to interact with specific molecules and then go back to being flexible. Another question, how do IDPs avoid being destroyed by the cell? Most misfolded proteins get cleared away quickly, but IDPs are recognized as functional because they often contain special sequence markers that say, I'm supposed to be like this. Their disorder is not a mistake, it's a design. In fact, disorder is increasingly seen as an evolutionary advantage. Many IDPs are involved in signaling, immune response, and transcription processes that require fast changes, short lifespans, and flexible interactions. These proteins act as molecular messengers and connectors in a busy cell environment where being too rigid would be a disadvantage. So, while the old view of proteins required a tidy shape for function, IDPs show us something new. Sometimes being flexible is more powerful than being structured. They break the traditional rules of protein science, and by doing so, they expand what we understand about life's machinery. Intrinsically disordered proteins, IDPs, lack a fixed three-dimensional structure, yet perform critical cellular roles by remaining flexible and adaptable. Their unfolded regions allow them to bind multiple partners with high specificity but low affinity, enabling dynamic signaling networks. This structural plasticity facilitates rapid conformational changes upon post-translational modifications, tuning activity in response to cellular cues. Disorder also permits regions to act as entropic springs or flexible linkers, spacing functional domains optimally. Evolution has conserved disorder in proteins involved in regulation, transcription, and signaling, underscoring its functional indispensability. The tumor suppressor p53 contains disordered activation domains that interact with myriad cofactors to regulate cell cycle checkpoints. It's like a Swiss Army knife. Its foldable tools can adapt to many tasks, whereas a rigid single-purpose tool cannot. IDPs often form fuzzy complexes, retaining disorder even in the bound state to accommodate different binding partners. This contrasts with the lock and key model, allowing one disordered region to recognize multiple targets by adopting distinct transient structures. The energetics of binding can involve a disorder-to-order transition or remain partially disordered, providing both specificity and reversibility. Such versatility is essential in hubs of protein-protein interaction networks, where signal integration and propagation require adaptable interfaces. Fuzzy binding also enables cooperative assembly and allosteric regulation through weak multivalent interactions. The disordered tail of the transcription factor CREB binds several coactivators by forming different helical motifs upon each interaction. It's like mud that can fill any shaped mold. Once formed, it sticks, but can easily detach or reshape for the next mold. IDPs maintain high conformational entropy, which can be harnessed to drive binding and assembly processes. Upon binding, the loss of entropy can be compensated by favorable enthalpic interactions with partners, fine-tuning the thermodynamics. Cells exploit this sensitivity by controlling local concentrations or modifications to switch interactions on or off. The entropic contribution also endows IDPs with rapid association-dissociation kinetics, vital for transient signaling events. Thus, disorder provides a built-in regulatory mechanism that structured proteins lack. Cyclin-dependent kinase inhibitors are disordered until phosphorylation triggers their binding to cyclin CDK complex, halting cell cycle progression. It's like a slinky toy. It's stored to randomness, springs it into shape when needed, but lets it collapse again without effort. Flat-folded regions are enriched in sites for phosphorylation, acetylation, and ubiquitination, enabling multi-layered control. The absence of a rigid fold makes these sites solvent accessible and kinetically favorable for modifying enzymes. Sequential or combinatorial modifications create codes that modulate binding, localization, and degradation. Disorder thus serves as a scaffold for complex regulatory logic, allowing a single protein to function in diverse contexts. Without a stable fold, IDPs can integrate multiple signals in a compact sequence. The disordered tail of RNA polymerase II is extensively phosphorylated to regulate transcription initiation and elongation. It's like a versatile control panel with many switches. Each toggle changes behavior without rewiring the entire system. IDPs often drive phase separation into membraneless organelles by engaging in weak multivalent interactions that nucleate dynamic droplets. Their flexible, repetitive motifs create networks of transient bonds, concentrating biomolecules and enhancing reaction rates locally. Disorder also prevents irreversible aggregation, maintaining droplet fluidity and responsive phase segment compartmentalized cellular processes such as RNA splicing and stress response without lipid membranes. Thus, disorder participates directly in spatial organization of the cytoplasm and nucleus. Fusin TDP43 proteins form liquid droplets in the nucleus via their disordered prion-like domains, organizing RNA processing bodies. It's like oil droplets in water that coalesce and separate reversibly, creating distinct microenvironments for reactions. Because IDPs function through patterns of amino acid composition rather than precise folding, they tolerate high sequence variability. Insertions, deletions, or substitutions that would destabilize a structured protein often leave IDP function intact or even enhanced. This evolutionary plasticity allows rapid adaptation to new regulatory networks or environmental challenges. It also explains the enrichment of low-complexity regions in signaling proteins across eukaryotes. Thus, disorder offers a playground for evolutionary innovation. The disordered activation domain of P53 shows considerable sequence divergence among species while preserving its regulatory role. It's like a flexible recipe where you can swap ingredients yet still produce a dish with the same overall flavor profile. Traditional structural biology techniques like X-ray crystallography fail for IDPs because they require a single stable conformation. Instead, methods such as nuclear magnetic resonance and small-angle X-ray scattering capture ensembles of conformations. Computational modeling and single-molecule FRET further elucidate dynamic landscapes. These hybrid approaches reveal how disorder underpins function but also complicate drug targeting and mechanistic understanding. Overcoming these challenges is crucial for rational design of modulators. NMR relaxation experiments mapped the conformational ensemble of the disordered P27 kinase inhibitor to understand its inhibition mechanism. It's like photographing a swarm of bees. You capture many positions at once, but never a single static picture. Targeting IDPs demands strategies beyond lock-and-key inhibitors, focusing instead on stabilizing transient structures or disrupting critical interactions. Small molecules and peptides can bind to disordered regions, shifting equilibrium toward less functional states. Alternatively, modulating phase separation by altering IDP interactions offers new therapeutic avenues for neurodegenerative diseases. Such approaches require screening for dynamic binding events and understanding ensemble behavior. Success in this area could unlock treatments for disorders linked to disordered proteins like ALS and cancer. Small molecules binding to the disordered N-terminus of c-Myc inhibit its interaction with transcriptional coactivators, reducing oncogenic activity. It's like tweaking a tangled string by clipping a few key loops. Rather than weaving a new knot, you subtly change its overall shape. Key mysteries remain about how IDPs balance flexibility with specificity and how cells prevent pathological aggregation. The interplay between disorder and ordered domains within the same protein requires further elucidation. Advances in in-cell biophysical techniques will shed light on IDP behavior in native environments. Understanding the full scope of disorders' roles promises insights into signaling, development, and disease. Ultimately, unraveling the IDP puzzle will redefine paradigms of protein function. Ongoing single molecule tracking in living cells reveals how the disordered region of P53 searches the genome for damage sites. It's like mapping the flight paths of hummingbirds. Tiny, rapid, and unpredictable, yet key to pollination and ecosystem health.